Cell communication is important as it allows cells to coordinate their activities, respond to changes in their environment, and adapt and survive. Without this communication, cells would not be able to adapt to dangerous situations and would die. We chose to explore the cell signaling pathway of cholera, specifically how it targets the epithelial cells of the intestines. And we chose to explore this pathway because two of our group members are interested in the medical field, and understanding diseases and how they work is an important aspect of this field.
Today we'll be talking about cholera, a bacterial disease that spreads through contaminated water, specifically about its cell signaling pathway and how it impacts epithelial cells within the intestines. Cholera utilizes a G protein-coupled receptor cell, or GPCR, signaling pathway. Before we talk about cholera, it is important to describe how GPCR signaling pathways work. In a normal GPCR pathway, a ligand, or signaling molecule, binds to a GPCR within the cell membrane, which then converts the extracellular message into an intracellular message. This happens in the form of a G protein, which is activated through the displacement of GDP by GTP. This G protein then activates an enzyme. In certain cells, this enzyme produces secondary messengers, which amplify the message through phosphorylation cascades. This will eventually lead to some cellular response. In a healthy cell, this process will eventually terminate as the alpha subunit of the G protein will hydrolyze the GTP into GDP, rendering the G protein inactive.
In the epithelial cells in the intestines, the presence of the normal ligand causes the activation of the G protein, which activates an enzyme referred to as adenylyl cyclase. Adenylyl cyclase then converts ATP to cyclic AMP, or CAMP. CAMP then activates protein kinase, or pKa, which is a protein responsible for the phosphorylation of other proteins. In these cells, protein kinase is responsible for phosphorylating a chlorine pump and a sodium channel, activating the pump and inactivating the sodium channel. This is done to expel waste from the body. Cholera causes a change in the cellular pathway by binding to the GPCR responsible for it. Once it has bonded, a nonpolar section of the cholera toxin that is capable of crossing the cell membrane is passed into the cell and binds to the G protein, changing its structure so the alpha subunit cannot convert GTP into GDP. Since the presence of GTP is responsible for the activation of the G protein, this forces the G protein to be constantly active, preventing the shutdown of the pathway. Because of this, there's a constant production of CAMP, and in turn, constant production of protein kinase. Due to the phosphorylation cascade and the amplification of cellular messages, this means that the amount of molecules that are pumped and kept out of the cell are enormous. This forms a concentration gradient that leads to massive water loss. This massive water loss within the intestine, combined with the amount of nutrients that are pumped out of the epithelial cells, results in diarrhea. If untreated, this leads to massive dehydration and death.